Okay, you guys can clearly see that I am not here, but there are some important notes that you guys need to have. So we are going to open Note Taker. And I'm going to start a default blank doc, and I'm going to call these Central Dogma Notes. Okay. If we want to look at the central dogma, we can summarize it in one like mantra. If we're talking about dogmas, it kind of makes sense to talk about a mantra, like something you can chant. It goes DNA to RNA to protein. And that's the sequence that all living things take to create proteins from a DNA sequence. All right. It's different between prokaryotes and eukaryotes. You're going to need to know the difference between prokaryotes and eukaryotes. Prokaryotes are our organelles or organisms that don't have membrane-bound organelles. They only have one circular chromosome. Right? They have no, don't need to yell at you, membrane-bound organelles. Because they don't have any membrane-bound organelles, they also don't have a nucleus. It's something called a nucleoid. Not essential, not nucleoid, not nucleotide. Okay. That sequence that we talked about at the top is a little different for them. They go from DNA to something called mRNA to the protein, which is also known as a polypeptide. These guys are different, and it won't make much sense until after we get done with the eukaryotes. They have no pre-mRNA. Again, it's not going to make much sense to you yet. But it literally goes DNA to mRNA to protein or a polypeptide. There's no in-between step, which we're going to look at. If you look at eukaryotes, however, it's a little more complicated. So in eukaryotes, you go from DNA to the pre-mRNA to mRNA to protein, which is still a polypeptide. All right. It does it in 2 steps. It happens in 2 steps in prokaryotes too, but we're going to kind of focus more on the eukaryotes. The first process where we go from DNA to pre-mRNA is called transcription. Okay. Transcription takes place in the nucleus. Because the DNA can't leave the nucleus. The DNA is locked in the nucleus. With the DNA locked in the nucleus, something needs to get that DNA code for making proteins from the nucleus where it exists to the ribosomes where the proteins get made. And this process of transcription allows that to happen. Okay? Transcription produces mRNA, which is messenger RNA. It's called messenger RNA for a reason. It is the messenger that carries the DNA message from the nucleus to the ribosomes. Okay? An enzyme known as RNA polymerase is responsible for making mRNA. Polymer, RNA is a polymer made up of nucleotides. RNA polymerase is the enzyme that does that. Okay. How does it know where to start? On the DNA sequence, there's a sequence of nucleotides called a TATA box, because it's literally the nucleotides TATA on DNA. That's called a promoter sequence. That tells RNA polymerase where to start transcribing. That's the signal. Hey, polymerase, RNA polymerase, this is where you need to start. Okay. The DNA strand that is transcribed into pre-mRNA is called the template strand, just like it was when we were looking at replication. So at least some things are consistent. The pre-mRNA is going to be edited in the nucleus. When it's transcribed from DNA, there are parts of that DNA sequence, and then therefore parts of that mRNA sequence that don't code for part of the protein. So those parts that don't get coded get pulled out, and then the pieces that are coding for something get put back together, okay? And those are the introns and exons. Introns are removed from the pre-mRNA. The exons stay in and eventually leave the nucleus. And there's a really good picture. It's from the CP book, but it's such a good picture that I need to show you. Okay. So there's the picture. Let me get into ink mode so I can draw on it. There's your DNA. It's got parts that code for proteins, for the genes, parts of the code that code for the protein, the important parts of the gene, and then parts that don't. Okay, you don't need to worry. I mean, we will talk about the cap and the tail at some point, but so the DNA, the introns come out, the light blue, and the exons stay in. So there's your completed mRNA. All right, that is transcription. That is the first part. The second part we're going to talk about is translation. Translation is harder, not necessarily harder. It's more complicated. Okay, translation. Zoom in so y'all can see. Translation means going from mRNA to protein. And because it's protein synthesis, it takes place in the ribosomes. And just like we talked about last class, depending on where that protein is destined will determine which type of ribosome it is synthesized in. So in a free ribosome, the protein will stay in the cell. And if it's synthesized on a ribosome on the rough ER, the protein will be packaged and secreted from the cell by exocytosis, but I'm not going to write that again. Okay. Let's talk a little bit about ribosomes, shall we? Sure, Mrs. Grady. We talked about this the last class. They are made of proteins and something called rRNA, which is ribosomal RNA. That's the only place ribosomal RNA exists, is in ribosomes. That's why it's called ribosomal RNA. We talked last class that it's made of 2 subunits, large and small. This is new. Ribosomes read the mRNA, mRNA in 3 nucleotide sequences called codons. You need to know that word, codons, okay? And every protein starts with the codon AUG. We call that the start codon. That signals the ribosome, hey, now I have to make a protein. Oh, look, there's AUG. Let's make a protein. All right. There's another kind of RNA, tRNA, which stands for transfer RNA. So the code has come from the nucleus through mRNA, and it gets to the ribosome. The ribosome reads the code on the mRNA. How do you get from mRNA to protein? That's where tRNA comes in. tRNA has the anticodon that is complementary to the mRNA codon. So it's the match. Like we made matching strands of DNA, complementary strands of DNA, we make complementary strands of RNA too. tRNA complements the mRNA. tRNA brings the right amino acid to the ribosome by matching codon to anticodon. That codon, anticodon matching, helps to be able to type, will continue until the mRNA reaches a stop codon. Not stoop, that was funny. Just like there are start codons, there are stop codons. It tells the ribosome, oh, okay, we're done. This is the end. Okay. Once that happens, the ribosome will split apart, and the protein will go for further processing. And the rest of the structures, you know, all that stuff. Okay. We know this process is not perfect. When there is a mistake in the code, we call that a mutation. And mutations happen fairly frequently. We talked about inversions and insertions and deletions and all that stuff, translocations. We're going to talk about the 2 big kinds of mutations that you can see. The first is called a point mutation. Point mutation is an alteration to 1 nucleotide base pair of a gene. So there's 1 letter that gets messed up. Just 1 letter. Like it substitutes. So instead of a U or a T, it's some other letter. That can result in one of 3 options. The first is a missense mutation. And a missense mutation is when a codon alteration will produce an amino acid, but not the right amino acid for the sequence. So you get an amino acid, but it's not the right one. The second kind you can have is a nonsense Not a nonsense is when changing the nucleotide changes the codon to a stop codon. There's still mRNA after it, but that stop codon stops the production, the continuation of the translation of the protein. Like, it's not going to happen. So anything that's beyond that, it's done, which can cause some big problems. And see, the last kind is a silent mutation. And that's when the mutation does not alter the amino acid sequence. And I'll show you how that can happen in a second. I can type. Good. The second kind we can talk about is something called a frame shift mutation. These are a lot more significant. Right, insertions or deletions can lead to larger changes in a polypeptide by causing a shift in the ribosome's reading frame. The 3-base sequence. So you put in another nucleotide or you pull out a nucleotide and the whole reading frame, that whole 3-codon sequence shifts. Okay? And it can cause some pretty major problems. I want to show you guys one of the coolest figures in all of biology. This is the codon wheel. Bum, bum, bum. You're going to see this again quite a bit. I'm going to use this in a lab after break, right? So the way you read the codon wheel is you start here in the middle, because codons are 3 bases. You start here in the middle and then work your way out to figure out what codon in mRNA codes for the amino acid. So if we want the amino acid, we want the codon UAU. Okay. You start in the middle at the U, you go to the next ring out to the A, and then you go to the U. And that's tyrosine, which I happen to know is abbreviated TYR. Okay. But if you had UAC, let me erase that so you can see it. If you had UAC, it would also be tyrosine. So that's how you can get a silent mutation. If that UAU became, got mistakenly replaced, that last U got replaced with a C, you're going to wind up with the same amino acid. Okay. If that became UAA, it would be a nonsense because it turned it from an amino acid that coded for, a codon that coded for amino acid, it turned it into a stop codon. All right. Don't freak out, don't panic. This is the first time we've seen it. I promise you we are going to go over it again. Have a great spring break, guys.